Hello, I'd like to talk with you today a little about the effect of alcohol on the immune system. I've primarily been doing research in immunology for a number of years, and then I got interested in toxicology, and that is a field that the study of alcohol falls into, and I got interested in the effects of alcohol on the immune system. One of the interesting things about alcohol and what it does to the immune system is we've known about it, or at least we've had some hint of it for a very long time. You notice this inquiry here, a very old document. It's into the effects of ardent spirits, which are just distilled alcoholic beverages, on the human body and mind, an account of a means of preventing and remedies for curing them. Well, Dr. Benjamin Rush was the Surgeon General of the Continental Army. That was before the US was even a country. And he must have been a very keen observer, because he discerned that there were a number of adverse effects of alcohol. And one of the ones that he wrote about in this monograph was that alcohol caused or enhanced the frequency of certain diseases, particularly pneumonia. And this was something that had not been documented by a scientific study at that point. So he just apparently was a good observer. It's interesting to point out, though, that in the past, casual observation, anecdotal evidence has not really served us very well. A lot of the research that I've done, and a lot of the research that many others have done on the effects of alcohol on the immune system and many other topics, use animal models for human disease. And one of the reasons for this is that there are certainly ethical issues that limit the types of experiments that we can do with human subjects. So there are, of course, also limitations on animal experiments, because we're required to use humane treatment of these animals. And there is still, though, much more flexibility doing experiments with animals and much more ability to reproduce findings than experiments with human subjects. So the experiments that we designed, their purpose is to reveal some generalizable finding. In other words, we want to be able to apply what we learn from animals to humans, and we want to be able to apply, in many cases, one situation to another situation. To generalize just collections of random observations are not sufficient. We really need to have a hypothesis in mind as to how things work. And then we do experiments to try to establish if we've got that hypothesis right. And then if we do, that gives us a way to understand causes of the observations, not just the observations themselves. And if we understand the cause, then we can know if we can modify the cause. We can also probably modify the effect or the outcome. And usually that's an adverse effect of a disease or of a exposure to a toxic material such as alcohol. So I've got a surgery book that was published in 1849, and it has a very interesting approach to medicine. What it shows me is that the anecdotal approach, the situation where we just do observations and we draw some inferences from our casual observations, that didn't work very well. And I want to show you some things that are a little bit amusing about one example of that. So here we have a medical book explaining to us in 1849 how we should treat acute tetanus. You're familiar with tetanus. That's why we get a shot when we get a puncture wound on our body. It fights the toxin that tetanus microorganisms produce and release into the circulation, and they cause all sorts of adverse effects, paralysis being the most serious of those. So we know that this is a disease caused by bacteria, generally secondary to some sort of a deep tissue injury. And we know what can be done to treat it now. And of course, the best way to treat it is not have to treat it. The best way is to avoid it by a vaccine. But here's what they did back then. They would bleed the individual. And by bleeding, they bled them sufficiently, removed blood such that the person was actually become faint. And the reasoning was, this was based on a rational, believe it or not, a rational thought. And it was that there was something in the body that was making the person sick. There was some sort of an evil vapor or something. And by bleeding, you'd get that out. And so bleeding was a very common treatment. You notice it's listed first here. So if you got tetanus, take out a lot of blood. That's got to help. Mercury. Well, of course, we all think of mercury now as poison. There are actually several chemical forms of mercury, some of them not all that poisonous, and some of them were used in medicine for a variety of purposes for quite some time. Calomel is the one they refer to here, but I can tell you it doesn't do anything to help tetanus. Purgatives, in other words, okay, we get rid of blood to get rid of the bad disease entity. They didn't know what was causing the diseases at this point. So let's get them out of the blood, but also let's cause the person to throw up endlessly and try to get them out that way. And so that was another approach that was very widely used. Tobacco, and you're thinking, oh, you make somebody smoke in order to get rid of a disease? No, you're thinking of the wrong route of entry of the tobacco. This was a tobacco enema. And the details of it are that the tobacco solution was given as an enema, and it was left in the person until they started having tremors and they became cold to the touch. In other words, right up until the point where they were suffering from nicotine poisoning, and then that was supposed to help. Cold is another thing that was thought to be useful. Tonics, iron, quinine, those kinds of things. And then it gets to the point where you've treated this patient with lots of different things, probably made them considerably worse than they were to begin with. And now you start the things that really aren't going to help them, but make them care a little less. Opium, that was on the list. And the way this is stated is its undoubted efficacy in some instances. Well, anyway, as I said, it didn't help with the disease, but probably made the person not feel quite as bad. The next was marijuana, resin of cannabis indica, and probably for similar reasons. And then another key treatment regimen for many diseases back then, ardent spirits, the same ones that Dr. Rush was talking about. Try lots of liquor. And again, same sort of approach I would imagine is that the person isn't going to get any better from the disease, but they won't be as concerned about it. And then nutrients as at the end. Well, of course, we know now that none of these things help tetanus, and probably almost all of them were harmful, but yet the medical community worldwide convinced themselves that these were good treatments. So I think a lot of times people don't have an appreciation for how much the methodology of science has done to make our lives tremendously better, and medicine is just one example of that. So Dr. Rush, as I said, though, must have been a pretty careful observer. He may have even kept some records and realized that he saw a lot of pneumonia cases in people that were problem drinkers. And so now carefully controlled research studies have shown that bacterial pneumonia and several other infectious diseases are more frequent and more severe in alcohol abusers than in moderate drinkers or abstainers. There's especially a strong increase in risk of infection in burn patients. And that's particularly problematic because a high percentage of people with severe burn injuries were drinking at the time they received the injury. It's a related set of events. So the person drinks too much, gets burned, and then the alcohol decreases the resistance of the immune system, or the ability of the immune system to resist the infections that occur after the skin is removed, which is one of the major defense mechanisms against infection. And then there's also evidence indicating that alcohol acts directly on key proteins in the immune system that are important in initiating a series of events that lead to resistance to infection. So alcohol is not a molecule that acts on every protein in the immune system. It tends to be quite selective. It acts on several, but not on all of them. So it does make sense for us to try to understand which things it's acting on more, and whether or not those can be addressed. I really like this picture. I wanted to show it to you. This is results from an animal experiment where we treated mice with a bacteria in the abdominal cavity. We injected them in the abdominal cavity with the bacterium. And then the mice on the left, the set of mice on the left, did not receive any alcohol. The ones on the right had alcohol a little bit after the bacteria were given. And this is 18 hours later. And what you can see on the left side is that there's just a few bacteria. I've pointed the white arrow there to one of the bacteria that's left in this slide. Those are little purple dots, are bacteria, but they're almost all gone. So the immune system has worked fine, and this mouse is not going to have any problem. In fact, it was showing absolutely no symptoms. It looked like a perfectly happy, healthy mouse. On the right side, you see that every one of those cells, and those are macrophages and neutrophils for the most part, white blood cells that migrate into the area where the bacteria are. And what you see is almost all of them are just full of these little purple dots. They're just full of bacteria. And so the immune system is really having some problems here. And if this was left untreated, these mice might not survive. So what are some practical lessons that we can get from a lot of these types of studies that have been done, both from animal studies and from studies with human cells? This is just some sort of practical points that we can infer from that big, actually large body of research. Relatively high blood alcohol concentrations are required to increase susceptibility to infection. So moderation really is advisable. The kinds of situation that occurred in that previous slide, those mice were given what would be a large binge type dose of alcohol all at one time. The lower the levels of alcohol that are given, the less that effect appears. And at some point, it just sort of goes away. So moderation really is important. Taking some of the precautions that we've all learned due to COVID-19 would probably be advisable after heavy drinking. Of course, we advise people not to drink heavily, but if people do, being careful about hand hygiene, being careful about being too close to people, especially if they're sick, have respiratory illness. Those are all things that would be very advisable. Also, no one should plan to drink or should drink heavily at or near a time of vaccination. Vaccination is just a situation where the immune system is taught to recognize a foreign substance, a microbe, and so that it can respond very quickly the next time it encounters that microbe. It doesn't learn very well if it's under the influence of alcohol, so that's a problem. Among the new drugs that are appearing most quickly, and some of the most exciting new drugs, especially for cancer, are immunotherapy drugs. These drugs work by manipulating the immune system to make the body's own immune system fight the cancer cells. I think it's clear that they're not going to be as effective if the person is drinking heavily during these treatments. There's not yet been time for a lot of research to be done on this, but based on what we know from the animal studies and from epidemiology in humans, I think it's fairly safe to say that will be the case, and it's a wise move not to take risk in that regard. So it may be eventually possible to specifically enhance immune mechanisms that we know are targeted by alcohol. There are a number of these that we know about already. And there is active research going on to specifically enhance the function of some of these immune mechanisms that we know are inhibited by alcohol. So in the future, it may be possible to anticipate what's going to happen. Alcohol is a very complicated subject, of course. Some people, some scientists even would say, well, the solution to that is to tell people to not drink heavily. And that is a solution. Unfortunately, we've been telling people that for centuries, and it's not an easy thing to do. As you know, alcohol dependence is very common. And once a person is involved, it can be very difficult to stop. Also, binge drinking has become amazingly popular. Something around 18% of the population of the United States above 18 years old has had at least one binge drinking episode in the last month or so. So that's pretty remarkable. It means that there's a lot of people engaging in that behavior and probably causing significant number of cases of infectious disease related to that. Thank you for your attention, and I enjoyed visiting with you about an interesting topic that I think we're beginning to know enough about to actually make some recommendations that might enhance health. Thank you.